So we're going to, Lee has suggested that maybe in order to maintain the continuity of what Dr. Matt has presented, that maybe we talk a little bit about what it is to participate in research at the CHUM. So qu'est-ce que c'est la research sur CHUM? So Dr. Matt has talked to us about a little bit about the biobank. So we want to tell you about what this is, about the opportunity that it is, and it's an opportunity for all of us. Les besoins des patients de SLA, premièrement, c'est le bon soin. Oui, c'est le bon soin. Et les réponses à leur interrogation, toutes les questions que vous avez déjà posées, ça c'est l'important besoin de les patients. Toujours les patients et leurs familles, et les chercheurs aussi, nous voulons de trouver une solution, de trouver un remède, un cure, vraiment. Ça c'est le but de tout. Il y a une possibilité pour les patients de contribuer à la recherche de leur vivant. Et c'est vraiment possible. C'est pas, comme Jean-Pierre avait déjà mentionné, pas toute la recherche est juste de prendre un médicament expérimental. Ça c'est pas tout qui est impliqué pour. Ça c'est that's not your only choice impliqué dans la recherche. L'objectif des chercheurs ici, dans cette salle, et parmi le Canada, dans toutes les cliniques, dans tous les centres de recherche, c'est de répondre aux besoins des patients, c'est de trouver un traitement réel et un traitement très proche, soon, bientôt. Maintenant, il y a les technologies actuelles qui nous permettent de stocker et d'analyser les volumes importants d'informations. C'est des grands bases de données. Ça, c'est les biobanques. So, comment tous les patients peuvent-ils participer à ce type de recherche? How can everyone participate? Comme Dr. Matt a dit, il y a, pour les essais cliniques, il y a des critères d'inclusion et d'exclusion et c'est vraiment strict. Mais pour participer dans une biobanque, dans un registre, il n'y a pas de critères. C'est ouvert à tous les patients. So, by accepting to be part of a research endeavor like this, basically this is what it entails. Is you're agreeing to provide access to your medical information in a centralized database in which it is completely de-identified, meaning it's anonymous. There's nobody in this room that would know that this file is associated with this person. There's nobody anywhere. It's a completely automatic, digitized sort of service. There's also potentially the opportunity to give biological samples to a central repository, and this is what we refer to when we talk about a biobank. So this endeavor is something that we've started at the CHUM that is going to be for all neurological diseases. ALS is our flagship disease. It's the one that many of us in the neuroscience group here in this building care about. It's one of I'd say the top three in this group. Researchers then have opportunity to have access to these databases from large populations of patients. And you can actually test the hypotheses and the causes and the treatments of the disease and how to follow the disease. And this is something that we talk about, we call biomarkers. So what is the biobank? This is the firm definition. It's a collection of clinical information and biological specimens that are donated specifically for the purpose of research. The goal is to improve our understanding of neurological health and disease and to eventually develop therapeutics. Pour tous les voies de signalisation qui étaient déjà impliquées dans la SLA et pour tous les nouveaux gènes et les gènes qui sont déjà établis, c'est nécessaire, c'est pertinent et nécessaire d'établir que cette gène, cette cible, cette voie de signalisation est vraiment une pertinence dans les maladies humaines. Dans le labo, on travaille sur le ver, le poisson, les souris. Moi, je travaille aussi sur les souris et les rats, des modèles d'SLA, mais aussi des cellules dans les pétris. Mais est-ce que toutes les choses que nous étudions dans le labo, est-ce que c'est vrai dans les échantillons des humains? Ça, c'est très important parce que finalement, c'est les patients, les humains que vous voulez traiter. For the biobank, I can tell you that it's a resource that has been thoroughly evaluated and approved by the CRCHUM Ethics Committee. This has been more than a year of paperwork and regulatory aspects in collaboration with Geneviève and others to really crystallize what is our goals. And our goals are to be able to invite patients to participate in the research in a different way than just taking medications. So what is being collected, we talked about, it's a collection of biological material, but it's not just that. So it's indigenous to your medical records, but they are digitized, as I mentioned. They're completely anonymized. And you can choose what you want to make available and what you don't want to make available. You can decide whether or not, as part of a normal blood test that your clinician has ordered, you can decide if you'd like to give an extra one, an extra vial of blood. That's the easiest example, which can then be banked for future studies. And of course, the power is being able to accumulate a number of these samples, right? Getting a population of these samples together so that our hypotheses and the medications that we're testing can then be validated in a large population of patients. For the most part, all of the samples that are collected align with what is already part of normal care. And as Dr. Matt said, you choose whether to participate or not. Declining to be part of research does not change in any way the care that one receives through the clinic. Okay? So what are the type of samples that comprises a biobank? So each type of repository has its own sort of list. This is our list of our ideal situation. Patients and family members can choose whether what they would like to donate. So whether it be an extra vial of blood, as I mentioned, or a small punch of the skin, which I won't lie, that it does hurt. But it's a small thing, it heals quickly, and from those type of samples, we can make what we call these induced pluripotent stem cells. So these are these cells that then can be reprogrammed to become the type of cells that are affected in the disease. And we can actually ask, in the context of what the patient's own makeup, of whether or not the mechanisms and the pathways of the drugs that we're studying actually work on those cells. In some cases also, a muscle biopsy, so a very small piece of muscle can be collected. This would normally be part of the diagnostic in ALS. It's not asking for anybody to volunteer for an extra anything. It has to be part of your normal care. A nerve biopsy, the same thing. It's a small nerve. I understand it's the sural nerve, which is not one that you'll miss so much, to put it that way. Yeah. I'm not a clinician, I apologize. And of course, then cerebral spinal fluid. So this is the fluid that bathes the spinal cord and bathes the brain. It's actually a really, really important sample because it's sort of the window into what's actually happening in the brain and the spinal cord, right? It's bathing it. These are the samples that are all possible to donate while you live with this disease. At the end, it is also possible to make donations for spinal cord and brain, but there is absolutely there's no pressure for that. It has to be something voluntary. It's as patients and their families would like. We're introducing this as this is all what is possible. Mais pourquoi participer à la biobank? For to participate activement in the research, you actually become really a part of the research that's actually happening. To find new genetic links, you can agree to be part of the genetic studies. And I will tell you, for those that are interested in that sort of type of research, the genetics is not communicated back to the family, right? It's all done anonymous. The labs do not know where the samples came from or associate with them with families. Everything is done with the utmost of confidentiality. You have the opportunity to participate in defining these new pathways, these new targets for drug development. To be able to help identify biomarkers. So biomarkers is something that gets a lot of press about. So what is a biomarker? A biomarker is anything that might give us some opportunity to diagnose the disease earlier, which would be great, because if we knew that, maybe when that treatment comes along, we can get it to the patient as soon as possible. The other is also as a prognostic prediction. So there are some patients that do well with the disease for 10 years. There's one that I met at the walk, and it's 10 years. And then there are others that, unfortunately seem to have a very rapid progression through the disease. What's the difference between those patients? Quelle est la différence? Ça, c'est très important. This is actually one of the biggest areas I think that ALS research worldwide is focused on. And of course, the ultimate goal, of course, is being able to develop new therapies. So, juste quelques exemples, c'est l'idée de Geneviève, c'est une bonne idée. C'est donne des exemples de découvertes faites grâce à des biobanques. Et le plus récent, c'est l'identification de nouveaux gènes NEC1. So see that Project Mine, it was all over the news in August. It was on every news channel. It was in every newspaper. There was a new gene identified in ALS, which is great because it's another piece in the puzzle of where we're going. Project Mine is a consortium. It's actually an international consortium in which countries from across the world have agreed to provide the DNA samples of patients. And so because the disease is rare, right, imagine it. I mean, I think it's 100,000 patients is the target overall. Canada has recently committed to 150 genomes, I believe, which doesn't sound like a lot, but it's a lot from a Canadian perspective because of our population base. The United States has agreed to donate 1,000. Belgium, I think, has done 1,000. Australia. Then there's also smaller countries like Brazil has made large commitments to generate about 100 of these to be able to sequence the genomes of ALS patients completely, so whole exome sequencing. And this is where NEC1 came from. And this comes directly from patients who have agreed to put a vial of blood in a freezer for researchers to look at later. There's also some interesting new stuff, and this is, I PubMeded this morning, is that there's this idea of being able to develop a classification system, so patients that are fast versus slow progressors that maybe develop ALS, and we know about bulbar onset versus limb onset, but maybe there's a way to develop a classification system that is completely an observational type of clinical research. And so it's basically agreeing to participate and allowing your information to be shared. Epidemiology in ALS, so we talk a lot about the genes, but we all know that most of the disease is not due to a genetic mutation that we know of yet, right? We call it sporadic ALS. We say that there are environmental components, but we don't really understand those. There are groups that are trying to figure that out, but I would say that it's because it's so unclear what those environmental aspects are. As a researcher, it's very hard to design an experiment to address those. C'est vraiment difficile. So epidemiology is a big word that basically says, is following kind of what's your family history, what's your lifestyle, do you smoke, do you exercise regularly, do you work on the farm, do you this kind of thing. That's the kind of thing that epidemiology is. It's a large push right now for ALS. ALS Canada has recently funded a postdoctoral fellow to look at this. They will have access to large databases that are from the States. And databases in Canada are not yet well established. This is also part of the registry that Geneviève mentioned. And of course, the big one is developing of biomarkers. So trying to find these things, because we can't biopsy the brain of the spinal cord, right? C'est pas possible d'avoir une pièce de la moelle épinière et des cerveaux. But the CSF, that material, that fluid that is bathing these structures, that is accessible. And then maybe there are things in there that can tell us what's the difference between the patients. Better, is there a way to be able to show whether these new treatments that are coming, that they're working, that the target is being engaged, it's called target engagement, that the mode of action, that we think that those drugs should be working. Is there a biological readout that proves it is working? Often with the clinical trials that have been done, the sad part is that the drug worked great in a mouse, usually, fish and worms too, but it worked great in these model organisms, and in the end it gets to the patient, and it's never really sure whether the target that we studied so much in these rodent models, if it actually worked. Did we actually get the target? That's what's actually quite cool about this timazide study, because they have a way to look at target engagement. So I want to finish sort of before I hand over to Alexandra and Lee that this is really a collective effort. Just in this in the University of Montreal network and the CHUMs, we have 4 researchers that are here in the room with us. We have clinicians, so Geneviève and Dr. Bottez are part of this work, but we all have Canadian and so national and international collaborations. We're not just this one little bubble of researchers in this little building in Montreal. We're part of a really large consortium of researchers across Canada. Canadian researchers are incredibly collaborative. It's really, really, really nice. It's an amazing field of work, and I did my 6 years in the States. It's not quite the same, okay? There's collaboration, it's true, but there are pockets. In Canada, there really is like a real national collaboration. There are lots of biobanks out there in the world, and Canada does not have a centralized one. And so this is sort of our effort to start this. So the MNI has been collecting for quite some time. Dr. Gange is very good at organizing the research at the MNI. Geneviève and I want to work together and get equally organized with the CHUM Clinic. And there is interest at the University of Calgary with a new study that's been funded. It's an imaging study, so it's MRI. But these patients are coming in as part of that study. It's very easy to add on, would you like to participate in a biomarker study or give a vial of blood for a future genetic study? So there is an organization that's happening to create a Canadian network, a Canadian repository. We do not have this yet established in Canada in a real central way. ALS Canada is considering to rethink, strategic funding sort of initiative to help establish these things. And we also have to say, probably it's a detail, but it's a really important detail from a researcher perspective. We plan to be able to collect the samples in a way that it won't matter whether or not the sample was collected at the CHUM Clinic or at the MNI Clinic, the University of Calgary, or somewhere in the Atlantic provinces. The idea would that be that the samples would be collected in a uniform way and they will be shared. Okay? This is the goal. It's always been the goal. And really, today was about engaging with you, the families, the patients, teaching you a little bit of science, teaching you what it is that we do in the lab, but also you're teaching us about what is the questions that are most on your mind. We've heard a lot about stem cells. I was kind of prepared for that, but that was a little bit more than I had expected. All the ALS patients I've ever met have been incredibly, and their families have been incredibly well educated about the disease and about the possibilities, very current on trials, always challenging us, big time, more than anything. So today is about trying to find a way in which we can work together. C'est une collaboration réelle. C'est pour la recherche, pour les médicaments éventuellement, pour les biomarqueurs, pour l'épidémiologie, pour les aspects génétiques, pour tout. So today I'm hoping is the start of something.